Hello and welcome to the H&R 19 notes on advances in genetics. You should be following along with your own copy of H&R 19 in your turn-in folder, and at certain points you'll be filling in some notes. So we've come a long way since Mendel and since the discovery of DNA, and genetic technology is now used in a number of ways. One way that you may be familiar with is in the context of crimes. We can use what's called a DNA fingerprint to identify a criminal based on DNA evidence collected at a crime scene. We can also use DNA fingerprinting to determine the paternity of a child. So if, for example, in here there's a mother and a child, and then four potential fathers, and what we can do is analyze the DNA and figure out who the father is based on how the banding patterns match up. These bands represent different alleles for particular genes. And so clearly you have to get one from mom and one from dad. Another way that DNA technology has been used recently is to identify old people, old important people, such as King Richard III. This is an article from last February where they found the remains of a skeleton. I believe it was near a parking lot somewhere in England. And what they did is they extracted some DNA from the skeleton, and based on other DNA banks they had available from relatives descended from King Richard III, they were able to figure out that it was indeed this king who had been missing up until now. So very powerful tools we have at our disposal. We're going to specifically talk about three main ways we can use advances in genetics to produce organisms with certain desired traits. And there are several key terms involved. You'll see them all highlighted in red, and I'll be filling in their definitions as we go. Okay, one main way that we can select organisms with certain characteristics is through what's called selective breeding. And the definition for you to fill in is that it's the process of selecting organisms with desired traits to be parents of the next generation. A classic example is the story of corn. Corn, as we're familiar with in the modern day, the plant produces very large cobs, such as this, and you can see the size of an average cob compared to that of a quarter. And obviously this is beneficial for humans because the larger the cob, the more food you get. But modern corn's ancestors from the wild, teosinte is the ancestor, the wild strain of corn, and teosinte produces corn cobs that look like this. And for scale, here's a quarter. We've come a long way from producing very, very small corn cobs to very large ones. And the way that this happened was primarily human selected, meaning that we took teosinte plants that produce larger than average cobs, and we bred those individuals together to increase the likelihood that their offspring would produce larger cobs. And we repeated this over many, many, many generations, and eventually ended up with the large modern corn cob. Another example, similarly, another produce plant, we have wild strawberries here, and through selective breeding, we have the modern strawberry that's raised in farms, much, much bigger. Selective breeding can come about using two techniques. One is called inbreeding, and the other is called hybridization. We'll start with inbreeding. The definition is the crossing of two individuals that have similar characteristics. So this describes our corn example and also our strawberry example. We were taking individuals that had similar characteristics, in this case, produced larger berries or produced larger cobs, and breeding them together with the hopes that we would get something similar over time. And the classic example of inbreeding that we're going to delve into later on is that of all the modern forms that dogs come in from their wolf ancestor, shown up here. And we can actually apply what we know about Punnett squares and inheritance to dissect an inbreeding example. So we're talking about labs here. They come in a variety of colors. They come in, I'm totally forgetting what this one is called. Golden lab, maybe? Then a dark fur lab, and then a chocolate lab, shown here. And so this problem says in labs, the allele for dark fur, capital D, is dominant over the allele for chocolate fur, lowercase d. Show a cross of a heterozygous dark fur dog with a chocolate fur dog. So we're not even considering the golden color right now. That actually happens with the introduction of a second trait, or a second gene. So we're just going to consider dark versus chocolate. So pause the video and fill in this Punnett square for this particular cross. Get your offspring. And then answer this question. Which offspring would you breed together if you desired chocolate pups? Meaning, which of the 4 potential combinations that we get would you breed together if all you wanted was chocolate pups in the end? When you're done with the problem, come back to the video for the answer. Okay, let's take a look. Here's the answer key for this problem. So we have our heterozygous dark, capital D, lowercase d, and chocolate. I don't even need to say it's homozygous because chocolate is recessive, therefore it has to be lowercase d. When you combine all of those alleles and their combinations, we get 2 heterozygous dominants, so these 2 are going to be dark, and then 2 chocolate recessive pups. And so if you were to breed any of these offspring together to get the highest percentage of chocolate pups you could, you would take, obviously, the 2 recessive individuals and breed them together. And that's how inbreeding occurs in dogs in general. You take the dogs with the traits you want and you breed them together. The benefits of inbreeding is that it helps us produce genetically similar organisms, specifically organisms with a specific function that's useful for us. So back to the corn example or the strawberry example, we were able to get food crops with higher yields of food. The cons is that it can increase the probability of genetic disorders. This becomes especially important with dog breeds, and our dog breed poster project will consider this topic specifically. You'll actually research a particular breed of dog and see what genetic disorders have come about as a result of this selective breeding. And when you think about it, this happens because many of these disorders are recessive, and so they're only going to arise when 2 heterozygotes are bred together over and over again. Here's a classic example of a genetic disorder from inbreeding or selective breeding in dogs, German Shepherd, with demonstrating something called hip dysplasia. See how the back slopes down and then the back legs are lower down than the front legs. This is a genetic disease where the hip joint is not functioning correctly. Very common in this dog. Okay, so we're talking about selective breeding. It can come about through inbreeding or through hybridization. Hybridization is the crossing of two genetically different individuals. So here, maybe you want characteristics of two animals that are quite different, and then you breed them together, and you get an organism that is a mix. Normally, though, actually not always, with dogs this doesn't apply, but with other animals, the offspring of these hybridization crosses is often infertile, which means that it cannot produce more offspring later on, so you have to keep hybridizing the two animals over and over again. So here's one example. We take a lab and a poodle, and we get a labradoodle, shown here. This is a case where labradoodles actually could breed with each other because technically dogs are all the same species and can breed. All the different breeds can breed together and have viable, meaning living offspring, and offspring that can have more offspring. This example shown here is a cross between a zebra and a horse. It's a zebroid, and a zebroid is not fertile, meaning that it cannot have little zebroid offspring of its own if you were to cross two zebroids together. However, the offspring are viable, meaning they live, they're healthy, and they look like this. And the idea behind this is that you're creating this hybrid to get the best traits or most desirable traits from both parents. Okay, so we just talked about selective breeding as one way to produce organisms with desired traits. Another way is to clone them. And this is where you produce offspring that are genetically identical to one parent. We talked about cloning in the context of asexual reproduction. So there are some organisms out there that reproduce this way all the time. Bacteria and other unicellular organisms, and also many plants can do this. Our potatoes being a perfect example. That potato is giving rise to an identical plant, identical to the plant that it came from originally. In mammals and in other complex multicellular organisms, cloning is not the norm. Normally, we have what's called sexual reproduction, where the offspring are a mix of the two parents. So cloning in mammals is extremely, extremely, extremely difficult. This picture here shows Dolly, the lamb. She was the first successfully cloned mammal. So this is Dolly, this is her mom. They are genetically identical individuals. Dolly was the result of, I can't remember the number off the top of my head, I want to say close to 300 different attempts at making this work. So it's very, very, very difficult to do. Okay, we have selective breeding, we have cloning, and then another way to get organisms with desired traits is to genetically engineer them. This is where stuff gets really wacky. So the definition of genetic engineering is when genes from one organism are transferred into the DNA of another organism. Classic example, go to PetSmart, some other pet store, you'll see probably these glowing zebrafish. And they don't naturally have this color. This color comes about because scientists have learned how to inject a gene from jellyfish, it's a glowing gene, into, usually it's a bacteria or virus. So they put the DNA into the bacteria or the virus. They then take the bacteria or the virus and insert that into the fish. And lo and behold, the fish will start expressing that piece of DNA, that gene from, in this case, a jellyfish. This is also very common practice in flowers. So this lavender rose, for example, this lavender color is not natural to the rose. It has been injected via a virus into the flower, and then that virus later incorporated that DNA for the purple color into the flower. Same thing with golden rice. And golden rice is cool because scientists have injected genes for the production of certain vitamins. I believe in this one it was I want to say vitamin D or C. I'd have to look back. And they use a virus to transfer that gene into the rice so that when you eat the rice, it's now fortified or enhanced with a vitamin that is essential to your health. And you can think about then giving this type of rice to people who aren't able to get these vitamins naturally in their diet. And for contrast, here's some normal rice, and here's the engineered rice here. And just to give you a visual, there's something called gene therapy, which you can think of as a specific case of genetic engineering where the end goal is to correct genetic disorders. But this generally also shows you how genetic engineering works. So here's the first diagram. Let's tackle this one first. We have the clone gene, normal allele. Okay, so here's the segment of DNA coding for a gene of interest. And we take that and we insert it into a virus. This shape here is a virus. This is the genetic material of that virus. And we can actually, in the lab, devise ways to take this gene and stick it into the virus. Then, step 2, we can infect the tissue, the target tissue, in this case it's a bone marrow cell, with that virus. So in goes the virus infects the cell, and in the process of infection, normally a virus ends up sticking its genetic material into the host cell, so that's just a normal process. Now, because we've injected this gene of interest into the virus, it goes bloop and inserts it into the chromosome of the host cell. And then we can take many of these cells, inject them into the patient. In this case, it was bone marrow, so these are cells down the center of your bone. And hopefully, we can get the expression of the proper protein from this new gene that we've injected into the bone marrow. You can imagine certain situations where you could have a bone marrow disorder because perhaps in this gene you had some sort of mutation along the way, and that mutation leads to some sort of sickness. And so by undergoing this process, you're attempting to inject healthy cells with the normal gene back into the person's bone marrow so that this person can start expressing the normal protein again. And this is used in a whole host of different genetic disorders out there. Hemophilia, a bleeding disease, that's one disease that they're developing this technique for. They've also used a similar technique to produce insulin on large levels, and insulin is a medicine used for diabetic people. This diagram over here just shows another example, only this time this is for the replacing a faulty blood clotting gene. So if you have hemophilia, you don't produce the normal blood clotting gene and protein, and therefore you bleed out if you get cut. But here we're putting the normal gene into the virus goes into the human cell, injects the normal gene back into the human cell, and then if you put these human cells into the body of a human with hemophilia, hopefully the body will start reading the normal gene and producing enough of these clotting proteins. Genetic engineering is an extremely controversial topic. It's not a black or white situation, good or bad. It has many benefits, but it also has concerns, so it's definitely a gray area. I just want to highlight some of these for you. Benefits from genetic engineering include being able to produce disease and drought-resistant crops. We have a growing world population. We need to be able to feed it all. This is huge. Being able to produce crops that are resistant to drought, that are resistant to disease. We can also use genetic engineering to produce foods that have essential nutrients. So back with the golden rice, that is enhanced with vitamin A, we can send that over to countries where there's a vitamin A deficiency and deliver that vitamin through a normal product like rice. We can use genetic engineering for gene therapy, where we're targeting certain genetic diseases by replacing the faulty genes. And then genetic engineering can also be used to produce useful materials. I mentioned this earlier. Producing insulin for diabetics, for example, was a product of genetic engineering. Concerns. Are these crops safe? The ones that we are genetically engineering. Do they have ill effects on humans when consumed? A lot of research has gone into that, and frankly, the jury is still out. There does not seem to be any clear indication that these crops are harmful, but people are still conducting research to figure that out. What will happen if genetically engineered organisms spread into the wild? This is a huge concern. If there is a strain of corn, for example, that's genetically engineered, that's really, really resistant to, say, disease and drought, and what if it escapes the fields where it's planted, goes into the wild, and then ends up displacing all of the wild corn or other wild plants that would have otherwise filled that niche in the environment? Is it morally right? Should we, as humans, be genetically engineering organisms in the first place? That's definitely a weighty question and something to consider. Finally, who owns these organisms? So if you develop the technique to, inject the glowing protein into, or the glowing gene into those fish, do you now own the fish? Or do you just own the gene? Or do you just own the process? It suddenly brings up the idea of ownership, specifically ownership over living things, and potentially even ownership of human genes. And this is a very hot topic, and recently the Supreme Court of the United States has ruled on several cases regarding this particular issue. Okay, so this is your time now to test yourself. I'd like you to fill in these 3 questions. You can fill it in the notes section or right here in the text. Pause the video. If you need to go back, feel free. And then when you return, we'll go over them. Okay, welcome back. 2 types of selective breeding. We had inbreeding, where we mated together organisms that were very similar. The main examples we saw with that were corn, strawberries, and dogs. We also have hybridization, where we take 2 dissimilar organisms and breed them together. We saw that with the zebroid and the labradoodle. There it is. What do we call an organism that has exactly the same genes as the organism from which it was produced? It is called a clone. We saw that with Dolly the sheep. What genetic technique has led to golden rice and other modified organisms? That is called genetic engineering. And when it is used specifically for combating genetic diseases, we call that gene therapy. Okay, that's the end of the notes. I hope you enjoyed and look forward to delving into greater depth with this issue when we learn about dog breeds.